I think we got some. There you go. That's good. Yep. In the jar.
It's canister 5, yeah?
Yeah. Thank you.
Yeah, thanks very much. That was a...
Please, onshore folks, mute your phone if you're not actively talking.
Was that 12? Yes, that was 12. Okay.
Mm-hmm.
12B and bucket 5. Okay.
Squid just hanging out.
And then before you guys speed off, we have a couple things we want to look at right in this view.
On the other side of the rock. Oh.
Yeah, right here.
Oh, I see.
Over there.
Great.
So, Stephanie, what I was talking about is like right below the squid arms. There's like 3 clusters of 10-centimeter branching or another.
Yeah, some dead coral of some kind. Yep, as soon as they stow the suction hose, we're going to take a peek at that.
Oh, yeah, we may have a little log jam in there.
Yeah.
So when we... can't believe that squid is just sitting in front of D2 on the bottom.
Yeah, he's one here.
Yeah.
Hi. What you guys doing? No, there shouldn't be any other buckets left. I think we're done with that. Yeah, that's fine. Bless you. Okay. That's fine. Can we dig it out of the hose when we get it on deck? Okay. Just wear gloves if you guys have to be the ones to go get it. That's my only recommendation. I don't. And we want to look at this dead coral before you run off. Whenever you get yourself set up. Okay. Sure. It's closer to the left, right? Yeah, it's like closer to the bottom, so more like 4 o'clock-ish. So yeah, if you keep spinning, it's coming into center screen. It's on the ground.
It's dead.
It's definitely dead. It's brown, gray colored. Now it's center screen, and 6 o'clock. No, it's a brown color. It looks like rock. It's a dead coral. I'm just trying to see if we can ID it.
Dead center at 6.
It's right below laser, yeah.
Yeah.
Take a snap zoom there, probably. Yeah, that's good. I think most of that's the same. Looks like it's covering most of this reef, actually.
And my screen is like freezing up.
Uh-oh.
I don't know if are you guys moving right now?
A little bit. We're bouncing around a little, but we're pretty much stable. They're going to zoom in.
Oh, it might be because I'm downloading about... Oh, that's probably it. I'm downloading about a 10-gigabyte file on my other drive.
That's what I'm
That's
after.
exactly what we're looking for.
John, it's impossible for me to tell what this is. All the corallites have been destroyed. It's definitely dead coral.
Heavily encrusted with sponges.
Yeah, it's been here a long time.
Some small polyps.
Yeah, I mean, it could be Lophelia. Could be Lophelia. I don't know. I can't
Yeah,
tell.
it's kind of stumpy.
It
Yeah.
looks Lophelia-ish.
Yeah, that's what I was saying this whole time. Looks kind of Lophelia-ish. Like, I really know. Because it's so beat up. So, yeah, that's good. My guess is it's probably the same species of the living stuff we just collected. Because some of the stuff isn't so bad. And then if you guys can look at 9 o'clock, there's some tufts in the sediment. Right past those rocks that we'd like to go check out. Look out.
Scott Francis says this could be, I think he said Okeanos when we were guessing what this might be growing in the sand.
In my pre-defense, in case it's not, I only mentioned that Okeanos grows in sediments. But I think you're right. I think this could be what it is. It should be very exciting.
Your guess is better than mine.
You like?
Full stop every time.
It looks like little Chrysogorgids.
Yeah, which is even better for them being Okeanos, because they look like little Chrysogorgia colonies. And there's several of them.
Each have a little associate in
They
them.
do each have an associate. That's so great. And they're all, associates are like all in the top center right.
In fact, Alan,
Oh,
I think
no.
you remember
Sorry. Right there. It's fine.
East End?
Go ahead, Scott.
I say, Alan, I think you remember Esprit East End?
Oh, yes, I sure do.
Yeah, so she was my PhD student after she was in Meg's lab, and she described a new species of Okeanos from out here in the Gulf. So this could be that new species. I don't remember. She she named two of them after each of her twin daughters.
Oh, nice.
Scarlet and yeah.
Very nice.
So there's 2 crabs in there.
So I'll look for the nodes.
I see nodes.
Oh, 100%. This is 100%.
Now, which twin daughter was it, or which daughter was this one?
Well, so Aurelia had a gold color to it, and Scarlatti had a red color to it.
Oh, I think this is a little more
red.
red. Yeah.
Yeah. But I need to check. One of those is only known from Norfolk Canyon. The other is from the Gulf. I have to check which one's which. And there is another species out here, Acanella arbuscula.
Mm-hmm.
So I'm not sure I can see it from this.
It looks like the rings are more of a gold color, but the polyps are pretty pink. Little mice and shrimp.
It's nodal branching.
You've got another Urotychus in here.
Urotychus. Thank you.
You've got another Urotychus.
Urotychus.
Thank you.
So this isn't in Chrysogorgia, but we have 1 bucket left.
Just to be clear, this is a bamboo coral, right?
I think so, yeah.
Yeah, it's a bamboo.
It's not a Chrysogorgia, but...
it is. Right, okay, I'm sorry. I thought you said it was a Chrysogorgia. I just
No,
wanted to
no.
be sure
No.
we're on the same.
No, Mary was interested in the crabs that lived in the Chrysogorgids, but this is not a Chrysogorgid, so I was checking to see if maybe Mary wanted maybe these anyway, or should we just leave them?
Sure, I want it.
Sure you want it. All right, guys, we have 1 bucket left, I believe. Is that correct? I mean, one bio box left? Starboard outer?
So this may also be Alan's opportunity to see the roots that he really wants to see. So if you're going to do it, you should just be able to up and out of the sediment.
Grab it by the base, is that what you're saying,
Just pick
Scott?
it up.
Yeah, grab it by the base and pick it up, and the roots should come out. And depending on how old it is, it'll be really impressive roots or not. We'll see.
Okay, so can we get this? Do you guys feel comfortable with that? It's going to be pretty hard. You're going to want to grab your claspers, but I don't want you to cut the base. I want you to pick the whole thing up out of the sediment if you can. So it includes the peduncle that's buried. It's really a root system, but and then really what we're targeting is those 2 squat lobsters that are in there. So the last time we tried this with the previous pilots, they just popped off. So you might want to, like, I don't know, do as little maneuvering as possible so that they don't go running away.
They didn't pop off right away, so they...
Yeah, it took a while.
Yeah.
Look, it took some
Just-
us irritating them.
But they
Yeah.
they did basically eventually decide running was better than staying.
And we have a full load today.
No, I just
Every
checked
box
the
is
paper.
full.
Acanella aurelia is the one from the Gulf, and as you pointed out, Stephanie, it is in partly reference to the gold-colored nodes, so this could be it.
Okay.
In which case, it might only be the 3rd known specimen collected.
Nice.
Check that. Take it back. It's known from a single specimen.
Nice.
This would only be the 2nd collection ever.
Wow.
And it's in the Keys, so...
Mm-hmm.
Yeah, this would be a new location. The previous one was from 800 meters in the northern Gulf of Mexico.
This Urotychus may be a different species from the one you tried to collect earlier.
Awesome. Hopefully they
Yeah,
stay.
Mary, I hope that's the case. Well, I guess we won't know because we didn't get the other one. But yeah, that would back up some of what I was suggesting earlier about possibly the host association. But as you say, there's so many of the Urticus that have been collected that don't have the host, so we don't have a good understanding of that. But when we go down with video, we often see them associated with a coral or a sponge, and so there is that suggestion that there might be choice among different species of the crab for what coral it's going to live in.
What's more interesting is you rarely find more than 2 per coral.
Yeah, that is very cool. And I was noting earlier that on the Parantipathes black coral that we collected in the seamounts in the North Atlantic, the pair that we got there, the pair of Urticus that we would collect off of them, it was always one male and one female together. And I remember distinctly one other colony that also had a third really small one. We were wondering, could that be the offspring? I mean, I don't know if that seems logical given maybe dispersal of larvae away from it, but that was the observation.
If you review the image that we just shot, that larger one, there's a very small one right below it.
There's a tiny one. There's
the
actually
bottom
3?
right. Yes, I saw a very small one on the bottom right.
Roland is saying there's actually 3 crabs on this. One of them is really tiny and below the large.
May want to request another zoom if you guys want to see it.
Yeah, if you want to snap zoom real quick. Sorry. I'm delaying you. I'm sorry. Yeah. Roland, I think that might be a worm. Are you looking below him? Right there? I'm not really seeing him. Okay, it's okay. We're good. That's a good view.
Looking forward to this root structure.
Mm-hmm. Yeah, hopefully it's not ridiculously large. It'll fit in the bucket.
It won't be that long, but the question is there some chunk of coral just below the sediment, so there's actually a holdfast? I hope not. I hope it's a root. I want you all to see this root, and you'll see it really looks like dripping wax coming off of the stalk.
Oh, neat.
Regarding it to if there were young ones living with the older ones, we know absolutely nothing about whether they have larval stages or not, and if so, where do they settle and how.
Sure.
I love it when I hear scientists have a sentence that says we know absolutely nothing.
Yeah.
It's so exciting.
For now.
That's right.
For now, exactly.
You should talk to more people who work on deep-sea crustaceans.
I think that'll work. Ooh, they look pretty active. Don't run away.
I know, don't run away. They're not. This is really... We may have to see the roots in the lab.
Mm-hmm.
It's really interesting, though. I keep thinking how we saw the rock pens, which are evolved from being, ancestors that were living in the sand to figuring out a way to live in the hard substrate. And this is sort of an example of the reverse.
Yeah, exactly.
Oh, there goes one of the...
Okay. Shh. Stay on there.
Kidding me.
Mr. Squid, you better move. No. Get away.
I think you dropped off.
I think he's still there. He's right here.
Good. Good.
I think
good.
we lost sediment, but
That's okay.
yeah. He's off. He's off. Yeah. All right. I didn't see him pop out, so that's pretty good.
Close them up.
That is 13B.
Yes.
Thank you. Want to grab a rock?
Sure. Do you have one in mind?
No. We filled up all our boxes, so.
I know.
That was super stressful.
That was exciting. It was awesome. Yes, that'd be great. Continue along the ridge and see what else we can find in our remaining time on the bottom here.
Did someone just call in?
Of the Mitchell Escarpment.
I think somebody may have hung up.
Okay.
But I do want to say that our day-to-day science typically is not so stressful.
I know. Usually I'm just parked in front of a computer crunching data.
Stressful or just this exciting?
Yeah.
Also, day-to-day is not this exciting.
Stressfully exciting, Roland. And as usual, my hats are off to the pilots who make this look much easier than it actually is. It's amazing. A delicate little coral, more than a kilometer under the bottom of the ocean, and this huge, monstrous piece of engineering can delicately pluck it up and get it into a box. Well done.
A round of applause from the chat room.
There's a star.
Mm-hmm.
At 6 o'clock or 9 o'clock? 25 minutes or 2? 2.5. Okay.
No. 25.
25. Okay.
Nice.
Yeah, we have a sea star right there.
Mm-hmm.
Sure, yes, please. Goniaster of some kind. Again, this dead standing corals pretty much everywhere. Ooh, those are new. Those pink soft corals are Nephthyids and
Pseudodrypha.
Yeah, nice view of the soft corals here. I think this is what we saw before. This is a better view. That zigzaggy, all these zigzaggy fans around it. I'll get off soon because no doubt Chris wants to talk about the sea star. But that, I think, is that planar chrysogorgia that I referred to earlier, looking even more like Pleurogorgia. This thing that Steve Cairns was working on.
I would have called those Plumarella.
Collected, so good stuff.
Yeah. Chris Ma tells us that is Circeaster americanus. It's a predator of many different kinds of corals. Mm-hmm. This ridge has a lot on it. Oh, we also got to remember, we have one last DNA sample, I believe, right? Water sample? We'll just do it right at the end. Hopefully it's got a nice, rich fauna nearby.
There's a white coral at 6, if we can get a zoom on that. It's near the bottom of the screen at this point. Mm-hmm. The white hard coral, yeah. So that looks Lophelia-ishy. Maybe not. It's very white.
These are... It's all flanked by... That's an Anthropotherian, right?
Which one?
Is it? Just all around. What is that?
No, I think
that's just the...
Chrysogorgia.
Oh, that's the planar Chrysogorgia.
Yeah, I think these are planar Chrysogorgias.
Yeah.
So call it Caryophyllid. Okay, thank you.
But if you're sitting here and you can get a slightly tighter zoom, that'd be good.
On the Caryophyllid? I mean, on the...
Chrysogorgia.
Chrysogorgia?
There's more. There's some nice fans above us. I think are the same species. Better front view. Yeah.
Is that a large Anthomassus?
Mm-hmm.
You had another Bathypalamonella on that fan-like structure you saw.
Oh, nice. Thanks. The Palmanella.
Bathanomas swimming by.
Oh, nice.
I thought at first, that squid's swimming backwards. No, that's not a squid.
Is this the same one? The flat plate chrysogorgid?
Yeah, this is what I was looking at before. I just want to be sure that it's not a primnoid.
I would have called it
Here,
primnoid. I didn't even know there was something that looked like different than that.
So you need to get closer in?
Yeah, the one in the background. The one in the foreground, I think, is a chrysogorgid, but the one in the background, I think, is a primnoid.
Mm-hmm.
You can see how the little teeny tiny polyps just hardly don't touch each other.
Yep. This makes a really dense network.
Can you see if they're scaly?
Scaly.
The polyps, yeah. Do the polyps look smooth, or do they look like they have scales?
They look smooth.
Huh. Okay.
If it was, what's the difference? Which one's which?
The smooth would be the chrysogorgia.
Okay.
And the scaly would be the primnoid.
Primnoid.
Yeah.
Do I think
But everything else I've seen today about this sort of thing makes me think of the Pleurogorgia type of chrysogorgid.
Chrysogorgia.
So I guess that's what we'll stick with.
Okay. We're good, guys. Thanks. Looks like a lot of this is mostly standing dead coral. So most of the stuff we've seen already this dive, I think.
Is that all sponge?
The blue, yeah. Can we zoom on that blue right there as we're passing center screen?
Another hyalinema off in the distance.
There's something different. Yeah, it's blue sponge. Maybe. That looks a little different than the one we collected, doesn't it?
I think it is different, but
no more room.
No. Yeah.
We got everything we could get. That's neat though.
It is.
And then if you go full wide on the right, there's some other stuff. Yeah, past the screen. You just have to drift to the right a little bit. Yeah. There's something I spotted. Ooh, there's a squad of squid. It's coming into view right now at the lower right. There's like a yellow and then a chrysogorgid. And then the white coral. That whole chunk right there is interesting. This is what I was...
All of this is really interesting.
Yeah.
Chrysogorgia in there, orange.
Yeah, so snap zoom. What is this? Sponge, maybe?
Yeah, the yellow on the left was a sponge.
Then we got more scleractinian, which it looks Lophelia-esque.
There's Chrysogorgia.
Yep, Chrysogorgid, Anthomastus. We got a couple hexactinellids. Okay. So that's Caryophyllid. No, that's good. You can keep going.
Oh, maybe the sponge just to the left to get the yellow sponge. Just get a good close-up of that. Yep. Thank you.
Oh, I got a critter in the front. Yeah, it looks like hexactinellid or something. And a sedimented.
It is sedimented. I think it's more likely a demo sponge. But
it's got that matting on it.
So John Reed mentioned that blue sponge, likely a desmocellid.
Oh, we're good. Thank you. Oh, you could have kept going if you wanted to. Fine with that. That looks like human debris there, guys. Be careful. Right below your right foot, looks like could be human debris. Line of some kind, right beneath you. That's a different black coral, isn't it? Can you zoom in on that orange one dead center? Or 6 o'clock? Thank you.
This one looks like a Leopathes from a distance.
And John was more definitive about the Selenosmilia.
That's what he thinks it is, Selenosmilia? Okay.
As you could see the bifurcate tips.
I think that's what we've been seeing this whole time. It's that same
Mm-hmm.
same species. I call this Liopathes. Let's see. There's a couple of...
That's not Euryticus. That's probably Sternostylus or something related to it.
Sternostylus. There's squat lobsters. There's also a shrimp. Really pretty one. It's almost purple. And then another small squat lobster on the upper left and upper right.
And did you say what the name of this?
Liopathes?
Oh, Liopathes, yes. Liopathes. It's very shaggy.
Mm-hmm. No, we're good whenever you're done making pretty pictures. We're good.
I like that shrimp.
Yeah, he's purple.
The red tail.
Mm-hmm. There's also a crinoid. There's still lots of this coral rubble, dead standing coral.
Mm-hmm.
It's been dead for a long time.
Yeah, fairly well eroded away and encrusted. And
Fresh dead is usually really white, like almost looks like a fresh exposed bone.
Mm-hmm.
And then you can still make out the calyces. You can see the little ridges in the calyces are all still intact. These have been here long enough to have eroded the calyce out. Usually ends up with just a little hole, so like a flare and then the hole in the middle, and then covered in that ferromanganese crust that takes a long time to form.
Do we have any sense of how long those time scales are? Like,
I don't know.
Not fresh as.
Yeah, I don't have any idea. I don't think anyone's ever done an aging study like that.
Mm-hmm.
It would be a cool study to do, but it might take 1,000,000 years to finish all your data. So, here's something fuzzy at 3.
Down in the crevice there. Looks kind of nice.
It's different.
Yeah, I haven't seen that.
It might be in a echinoderm. I think so. I think it's an urchin.
Yep, I think so too. Hadn't seen very many urchins really in general on this. We did make that one awesome collection of Habrosidaris, really rare.
Yeah, the little
urchin.
tiny one, right?
Mm-hmm. Beautiful animal. One of my highlights from the whole expedition so far, by far. Yeah, it really stands out to me. But I'm not sure. Do Do you recognize this urchin at all, Steph?
Nope. I mean, it's not a Sideris, I don't think. It's not really a pencil urchin. It's usually pencil urchins have a couple of really thick spines in there.
Mm-hmm.
So I wouldn't necessarily call it that.
It's a Okeanos.
Okay. Sounds good.
It's nice. Looks like it has an associate on it.
Oh, I think that's on the rock in front of it.
Oh, you're right. It's just right. Not climbing up its spines. It's on the rock.
You can see how really long some of his tube feet are. Look at there's one hanging out on the left.
Mm-hmm.
It goes well past the spines. We're good whenever you guys are. Thank you. Another Crassagorgid and stuff above us. Looks like Hylanema up in the sediment up there.
Yeah, we've seen a number of them in the sediment.
It looks like this was once a fan of some kind. A really thick stem that's died. So it looks like a branch. And then Bathopathes maybe.
And that's just nice hole. That hole had a nice
We'll swim through.
Yeah. We've got a tunnel. Stichopathes.
Yeah.
Another Okeanos.
So it does look like there was some around sturdy rocks, but a lot of this is dead coral rubble.
Mm-hmm.
Lots.
It just makes me wonder, how long ago this was, more, right?
I'm sure we can date it, right? I don't mean like take it to dinner. I mean like date the coral. Like you could get an age on it.
Yeah, you probably could with the skeleton. Maybe we got some dead bits in the hose when we knocked off that.
I'm sure we have some dead bits. Really interesting. Only 2 minutes of bottom time left. Yeah, there's a neat one. Can we look at the yellow fan at noon?
Sort of.
After the ellipse go away.
A little cone shape.
He just banged into the reef. Yeah, it's now closer to 3 o'clock. Yep.
Yeah, there's another star down there in the bottom right.
Oh, yeah, I see him. I'm looking at this yellow thing. Yeah. No, it's almost dead center. Yeah, it's right next to lasers. So if you were to snap zoom, it would be in the view. It's a hexactinellid.
It is another hexactinellid with zoanthids growing on it.
Yep, that's a big one.
Really big and robust.
Oh, man. Yeah, there's something in the back of
fish.
Fish.
Like a skate. I think a
Flatfish?
skate.
Yeah.
Or, maybe flatfish.
It might be as. Yeah, do you guys see that? If you can go look at him next. If it's a ray, it's just going to take off, so be careful. Snap zoom on that ray. Oh, well, we need a water sample before you take off, too, real quick. Yeah, it's a ray. Benthobatus or something. Sweet. Capture time. Thank you. Oh, no, it's a really spiny-tailed one. Okay, thanks. We're good. Thank you.
Yep, we can grab some water when you're ready.
I think they did it already.
Oh, did you? Thank you.
So that's sample 14. No. 13 was the Gorgonian. This is going to be 14.
That's great. Great. Sorry about that star. Oh, there was Bathynomus out there too. Someone spotted.
Really?
I did not notice that.
Oh man, we're coming up with a full load today. Every bucket is full, except for the rocks.
Except for the rocks. Mm-hmm. The rocks are always the hardest.
Ooh, that was a good dive.
That was a great dive.
All right, everybody. I haven't even done a recap. We had so much to look at, I didn't even recap today. Did you recap today?
No, we had no.
All right, we'll recap right now. We're 40 miles south of the Florida Keys on something called the Mitchell Escarpment, which is actually south of the Portales Terrace. 1,160 meter depth was our starting point at the base of this. I guess that's a wall.
Yeah, it was
pretty cool.
It was a wall, for sure.
Definitely a hill.
Yeah.
We ended our dive around 1,110, 1,105-ish at the top of it.
After going up the wall and then down the edge of the wall.
Right, and at the beginning it was really sandy, and then it became pretty rocky, more like that eroded white, looks like old reef rock. Mm-hmm. And then when we got to the top, we saw a lot more standing dead corals up there, and basically life the whole dive. There was lots of cool stuff. We saw multiple cladorhizids, which are carnivorous sponges.
Yes.
And we have filled every biotic bucket that we have on the ROV, every bio box we have on the ROV, and every water sample we have on the ROV. So that was a super successful dive. However, we'll be up until Saturday cleaning all this data.
It's going to be a busy evening, right?
Yeah.
It's going to be fun.
Make sure we remember to eat.
So I have a question about carnivorous sponges. Wouldn't all sponges be carnivores? Because don't they all eat, like, living matter?
Right?
They get the name carnivorous because they eat other animals. So most sponges will be eating bacteria or small eukaryote single-celled organisms.
Oh, okay.
And basically, filtering them on their little choanocytes or special feeding cell types.
They won't eat like copepods on the regular sponges?
No.
Oh, okay.
No, they really don't.
But the carnivorous ones would capture those?
Yes, small, mostly larvae, so they're eating pretty small crustaceans.
Got it.
Yeah.
That's cool.
Yeah.
And they're active, right? They actively capture that, or?
No, they have spicules that sort of act like Velcro, and so when they bump into them, they're just the larvae get locked onto them, and then very slowly the sponge cells overgrow the prey, and then they.
And then Scott was saying, I think, that the bacteria inside there is what dissolves the actual animal.
Yeah.
Oh.
And they just so happen to be able to take
that up.
So they have like their own
leftovers.
microbiome.
Mm-hmm.
Oh yeah,
that's cool.
Sponges are really big with microbiomes.
Well, I guess if anybody from DARPA is listening, we have a new Velcro for you out there.
And yeah, it's pretty interesting because they don't have any extra digestive enzymes and stuff.
Yeah, that's neat.
Mm-hmm.
So for a heads up, everybody that's listening, we will have our dive planning call at 3:45. And we will not be diving tomorrow because we'll be transiting back to the Blake Plateau region. But we will be planning for the next day on November 12th, and that will be the Knowles North region, which the mapping shows some really nice coral mound features, or what we hope are coral mound features, and hopefully lots of live coral on it, which we haven't seen much this cruise because we've been gallivanting in new places and out on the eastern Blake, which doesn't have as much coral and stuff. Hopefully you tune in and we can talk through it. See you in 10 minutes or 6 minutes.